morning. Today we are going to describe about structure of polynucleotide chain. In previous class we discussed about that polynucleotide chain structure. So before going to the structure, we had given that what is nucleotide, what is nucleoside. Nucleoside means nitrogen base is attached to the pentose sugar of first carbon by N-glycosidic bond, that called nucleoside. There is two types of nucleosides are there. In DNA deoxyribonucleosides, RNA ribonucleosides. Same to same. Then nucleotide means what is the nucleotide? Phosphate to sugar to nitrogen base, all three were there. Phosphate group is attached to 5' hydroxyl of nucleoside is called nucleotide. Then we'll see, before going to the polynucleotide structure, phosphate group is attached to 5' hydroxyl of nucleoside by phosphodiester bond. Phosphodiester bond is nothing but form the nucleotide. So this is very important. You see here, this is nothing but phosphate group. So phosphate group is attached to the 5' hydroxyl. Here may hydroxyl group is there. So when you take, this is exactly phosphate group is attached to the 5'. This is 5'. Phosphate group attached to the 5' of, in this side hydroxyl group there. So I can take like this. So phosphate group is attached to the 5' hydroxyl of nucleoside by phosphodiester bond. Then it is called a nucleotide. So we can see here, this is nothing but one nucleotide. This is nothing but one nucleotide. So this is one nucleotide. So these are the nucleotide structures. Of course, if phosphate is not there, then this is a structure only nucleoside. They go here. From here total it is if phosphate not there, remaining thing nucleoside. Now you see, phosphate is attached to 5' hydroxyl of nucleoside by phosphodiester bond. That is called a nucleotide. Then two nucleotides. Here one nucleotide, here one nucleotide. Two nucleotides are joints in this is also important. Two nucleotides are joints in a 3' to 5' direction. This is very, very important. 3' to 5' direction, then form dinucleotide. This is very important, Anna. So here, see here, two nucleotides. These two, this one nucleotide and this one nucleotide. For example, this is nothing but 5' here. This is nothing but 3'. This is the 5'. So 5' I said now. So this is the 3'. So two nucleus, another nucleotide will join here in 3' direction only. Another nucleotide, this is the nucleotide total. Here another nucleotide joins in the 3'. 3' already there, hydroxyl group, OH group, here OH group. So another nucleotide will join in 3' because 3' having hydroxyl group is present. That's why another nucleotide or anything will be joined here only. So that's why this nucleotide and this nucleotide, it joins a 3'. This is onwards 3' to 5' direction. Then form dinucleotide. More than two nucleotides joins, then form polynucleotide. More than two nucleotides are joined together, then form polynucleotide structures. Polynucleotide. So here more than one there, 1, 2, 3, 4, more than one type of nucleotides joins together, then form polynucleotide. So this is nothing but polynucleotide chain. Of course, a polynucleotide chain consists of two ends. A polynucleotide, this is totally polynucleotide chain. A polynucleotide chain consists of two ends. Then you see, this is one end, this is a one end. Two ends. First end is nothing but 5' end. 5' means carbon position 5. Here carbon position is third. So that is a third prime means carbon position third. Here 5 prime means carbon position 5. So at 5 prime end, 5 prime here 5 prime end always phosphate group is present. Third prime end is always hydroxyl group. A polynucleotide chain consists of two ends are there. One is a 5 prime end, another one is a third prime end. 5 prime end having always a phosphate group, third prime end having hydroxyl group is present. So because of hydroxyl group, third prime end, any compound, any other thing will be joined here only because OH is a functional group. That's why two nucleotides are joined in third prime to 5 prime direction only. This is the general structure. And one more important thing, then this is nothing but nucleoside without phosphate. The basic unit or backbone of the polynucleotide chain is phosphate to sugar. This is nothing but pentose sugar. Phosphate to sugar. Phosphate to pentose sugar. I mean deoxyribose sugar. Phosphate to pentose sugar is the backbone of polynucleotide chain. You continue. Phosphate to sugar. Phosphate to sugar. Phosphate to sugar. Phosphate to sugar is the backbone of a polynucleotide chain. While nitrogen bases, adenine, thymine, guanine, cytosine, nitrogen bases are joined to the pentose sugar towards inside. This is nothing but a polynucleotide chain structure. Even 3-mark question, very important. Briefly describe about the polynucleotide structure means you have to draw this diagram and some information. So I am going to repeat. See here, a polynucleotide chain consists, before going to that, a phosphate group is attached to 5 prime hydroxyl of nucleoside by phosphodiester bond, then called a nucleotide. This is a phosphate, here joins, then form, this is one nucleotide. Two nucleotides are joined in third prime to 5 prime direction, then form dinucleotide. More than two nucleotide joins, then form polynucleotide chain. Even in polynucleotide chain, two ends are there, one is 5 prime, another one third prime. 5 prime end having always phosphate group, third prime end having hydroxyl group. Because of hydroxyl group, they can ability to join any other compound. That's why another nucleotide, anything will join at a third prime end only. And then the backbone of the polynucleotide chain, phosphate to sugar, phosphate to sugar, phosphate to sugar, phosphate to sugar, pentose sugar, backbone. Nitrogen bases projected inside only, towards the inside. These nitrogen bases are attached to the pentose sugar. This is nothing but structure of a polynucleotide chain. Some additional information about RNA. So this is nothing but DNA. Now on RNA having additional hydroxyl group is present. RNA having additional hydroxyl group. Then see RNA structure. RNA having additional hydroxyl group. Second prime additional hydroxyl group present on second prime. So second prime, that's why it is a less stable, more reactive. Actually one hydroxyl group is there, then ability to join another. But here additional hydroxyl group present on the second carbon in RNA. That's why it is a more reactive, less stable. Not only that, in RNA uracil is present in place of thymine. In RNA uracil is present in place of thymine. Then one more word, thymine another name, 5-methyl uracil. 5-methyl uracil. Thymine another name is a 5-methyl uracil. So sometimes in questions also, in DNA, identify the correct nitrogen bases. Adenine, guanine, cytosine, 5-methyl uracil. All are correct. Kaiku, thymine in place of thymine, 5-methyl uracil. Both are correct. But only uracil present in RNA. This is nothing but additional information in basic points. Now we going to that next structure. History, next information. History of the DNA now. History of the DNA, genetic material. History of DNA, deoxyribonucleic acid. So history, what is history, sir? So who discovered first thing? According to NCERT textbook. So that point is important. So DNA, first type, DNA, history of DNA. You see here, history of DNA. Frederick Miescher. Frederick Miescher, scientist, Frederick Miescher, first identified or first found the acidic structure in nucleus. Acidic structure in nucleus in 1869. Then later this acidic structure he had given the name that is called a nucleon. Very important NEET question also. Who given the nucleon name? Frederick Miescher. Who discovered the first acidic substance in nucleus? Frederick Miescher in 1869. So Frederick Miescher discovered the acidic structure present in nucleus. Later he called it is a nucleon. Then next scientist, Wilkins and Franklin. These two scientists discovered the X-ray diffraction data, XRD short form, X-ray diffraction data. So Wilkins and Franklin discovered the X-ray diffraction data, which is useful for 3D structure of the DNA. To see that or to view the 3D structure of DNA, X-ray diffraction data is helpful. So X-ray diffraction data is discovered in the year 1953 only. 1953 XRD. Then Erwin Chargaff scientist discovered the Chargaff's rule or Chargaff phenomena. According to Chargaff rule in 1950, year important, 1950. So according to Chargaff rule, purines and pyrimidines are present in 1:1 ratio. 1:1 ratio. How much purine there? The same amount of pyrimidines are there. It is same amount. According to this concept, A plus G by C plus T or T plus C equal to 1. This is the formula. So Erwin Chargaff, according to Erwin Chargaff rule, A plus G by T plus C. Ag means purines. These are pyrimidines. Purines and pyrimidines are present in 1:1 ratio. 1:1 ratio. So Watson and Crick. So important one. Watson and Crick. In 1953 proposed DNA double helix structure. Who proposed the double helix structure of DNA means Watson and Crick. In which year? 1953. So Watson and Crick proposed that DNA double helix structure, DNA having double helix structure based on Wilkins and Franklin XRD and Erwin Chargaff rule. Sometimes NEET questions also. Watson and Crick proposed the double helix structure of DNA based on which concept? Option A, Wilkins and Franklin XRD. Option B, Erwin Chargaff rule. Option C, both A and B. Option D, no one. Both A and B correct. Watson and Crick proposed the double helix structure of DNA based on Wilkins and Franklin XRD X-ray diffraction data and Erwin Chargaff rule in 1953. For that he got the Nobel Prize in 1962. So Watson and Crick got the Nobel Prize in 1962. And one more scientist, Altman given the name nucleic acids. Additional information about this thing. Now, before going to the double helix structure of DNA, so what had given Erwin Chargaff? Erwin Chargaff given the Chargaff rule. Then according to this rule, purines and pyrimidines are present in 1:1 ratio. A plus G by T plus C equal to 1. Then you see here how to sometimes may chance to ask some question based on this concept. See Nana, in double helix structure of DNA, question Nana, in double helix structure of DNA, double helix structure of DNA, double helix structure of DNA, adenine equal to 18%. Then what is the percent of guanine. So in double helix structure of DNA, adenine they were given, but we need to find the guanine percentage. How? Sir, formula for this. So according to Erwin Chargaff, answer. According to Erwin Chargaff, A plus G by T plus C equal to 1. Then adenine equal to thymine. According to that, adenine equal to thymine. So adenine how much? Adenine is 18. So thymine also 18. So A plus T equal to 36 percentage. A plus T equal to 36. Then we need to find G plus C, guanine plus cytosine. Kaiku A plus T, adenine and thymine both are same. So adenine equal to thymine, thymine equal to adenine. So G plus C equal to 1. 1 means 100. 100 total percentage 100, and adenine, guanine, cytosine, thymine total you take the 100. So A plus T equal to 36. Then G plus C equal to 100 minus A plus T. So 100 minus how much? 36. Then how much it comes? 64. So G plus C equal to 64 percentage. Then G equal to C na. Guanine, cytosine same na. Guanine, cytosine same concept. This is we know. G equal to C. So 64 by 2. Then that is nothing but 32 answer. So guanine equal to 32. 7 cytosine also 32. Such type of questions also may chance to ask. Then did you understand? This is nothing but purines and pyrimidines ratio. Now you just verify that Chargaff rule. A plus G by C plus T equal to 1 I was given. Then verify the concept. A how much? 18. G how much? 32. Then C how much? 32. C also 32. Then thymine how much? 18. 18 equal to 1 na. So 18 plus 32, 50. 32 plus 18, 50. So 50 is to 50, 1. Verified. So A plus G by C plus T equal to 1 according to Chargaff rule. Now one more question about this. One more question. See here. According to that, then in double helix structure of DNA, guanine equal to 0.4%. 0.4%. Then what is the percentage of thymine? What is the percentage of thymine? We need to find that. So 0.4. Then find that. Adenine equal to thymine, guanine equal to cytosine. This is a basic concept we know. They were given guanine 0.4. Then cytosine how much? 0.4 only. 0.4. Then G plus C equal to how much? 0.4 plus 0.4? 0.8. Now you see, we need to find A plus T. Adenine plus thymine equal to 1 minus, here not then a percentage. So 0 points, that's why 1 only take. 0.8. 1 minus 0.8. So 1 minus G plus C. So that is equal to 0.2. So then 0.2, adenine equal to thymine. So 0.2 by 2. So then 0.1 answer. Adenine 0.1, thymine also 0.1. So for example, you check the verification. Adenine plus G by A plus G by T plus C equal to 1. This is the Erwin Chargaff rule. Adenine how much? 0.1. Then guanine how much? 0.4. Then thymine how much? 0.1. Cytosine how much? 0.4. Then equal to 1. 0.1 plus 0.4 how much? 0.5. So here also niche 0.5 equal to 1. So cancel 1. That is something but Erwin Chargaff rule. Such type of questions also may chance to ask here. Erwin Chargaff. So this is nothing but important information about the DNA history, whatever the requirement. So this is the maximum information.